All right. Good evening, everyone. Thank you so much for being here. Thank you to everyone that is joining us online. My name is Andrea Godoy Mendoza, and I am extremely happy to welcome you all here today. I have the pleasure of overseeing Roatan Marine Park's research program, and today we want to make use of this event, this space, and your presence to formally launch our Spawn Watch 2025 season. Now, I'd like to ask if you are at all familiar with the efforts that Roatan Marine Park has been carrying out for the past 3 to 4 years with regards to coral spawning monitoring. Raise your hand if you've ever been in the water with us. And people online, feel free to also react if you can hear me. Awesome. So we have some people. So basically today we are going to be inaugurating one of the most important events for our organization. Also one of the most important events for our coral reefs, which is the coral spawning season. Today we have a very action-packed session for you all. We have an interesting talk from a very important visitor. We also have very important guests here present. And we are also going to be presenting some important updates with regards to our Coral Restoration Center, Roatan. So extremely happy to have you here. We appreciate you all being here on time. We appreciate your presence. And without further ado, Dr. Robin Tyler Smith. He is the Associate Research Professor at Arizona State University, and he also has founded Coral Engineered Ecosystems and has been a crucial element in making our lab space happen very, very soon. So I would like for you to please welcome him today.
Thank you. Okay, let's see. Thank you for having me. Again, I'm Robin Smith. I'm Associate Research Professor at Arizona State University, but I'm based in Hawaii on the Kona side. So we have a facility there, and we run a program that's new. It's the Akoakoa Reef Restoration Program, and it's part of Arizona State University. Akoakoa is a Hawaiian word that it's a native word that has a dual meaning. So it means the coming together of community, but it also refers to coming together of coral polyps on the substrate to build the reef. So it's a pretty cool name for our program. But today I'm going to talk to you about my work there. I'm the lead scientist for sexual reproduction and restoration based on sexual reproduction. So I'm going to talk about sexual production of corals engineered for restoration at consequential scales. And there's a couple things in here that I'm going to focus on. Corals engineered for restoration, what does that mean? And then what does consequential scales mean? So mahalo in Hawaiian is thank you. And this used to be my last slide, but I gave this talk a couple months ago for ASU leadership that came to visit from Tempe. They came to the facility. We just commissioned the systems in September. So they wanted our team's strategic plan, like a 5 to 10 minutes of what are you doing as the component of restoration. I finished that talk with this slide, and now I start with this slide because what I found interesting was after the talk, nobody wanted to hear about all the graphs and figures and data that I painstakingly put together. They were just interested in the slide. So in Hawaii and probably here, it's very important. They call it talking story. So the Hawaiians don't like to just jump into business. They like to talk story. They like to get to know you. So I've kind of changed my presentation around, and I start a little bit with a little bit of story and a little bit of my background and myself. This is my son River. This picture, he was 6 years old. By 6 years old, he was already one hell of a free diver. He was getting down to like 30 feet. And this is a shot of him in the Bahamas, in the Exumas, swimming down to a big star coral that was part of my research for over a decade. River grew up kind of the same. He's 14 now. He grew up pretty much the same way I did. This is me when I was little. I was born and raised in Florida, 4th generation Floridian. I grew up fishing, hunting, spearfishing. I think I started surfing when I was 4. And then at 6 years old, every summer, my grandfather and I would jump aboard his little 24-foot sailboat. We'd sail down the west coast of Florida, all throughout the Keys, over through the Bahamas. I would take a month off of school. So 3 months every summer until I was about 14, I would spend with my grandfather on the sailboat. Every year we would go down to, as soon as we got to the Florida Keys, as any good grandparent would do, he's like, okay, where do you want to go? And my answer was always the same. I was always like, I want to go to Cary's Fort Reef. Cary's Fort Reef back then was one of the largest elkhorn stands in the Caribbean. I mean, it was literally hectares of elkhorn as far as you could see. And for me, at 6 years old, I mean, this was like magical. I would spend all day in the water. I'd swim through the channels. I'd only come back to the boat to tell them the next cool thing that I saw, and then I'm off again. And when he'd call my parents to check in, it was always funny. He's like, "Man, that boy spends more time in the water than he does on the boat." This place to me was literally my playground throughout my childhood. It was awesome. So fast forward to undergrad, and I got the displeasure of watching my playground pretty much succumb to death and a graveyard. That's Cary's Fort in 75. In 83, I think, it got its first bleaching event, wiped out a lot of it. 2004, there's not much left. And then from there on out, white plague disease and future bleaching events basically did it away. Millions and millions of dollars have been spent on restoration for this reef, and it's not coming back. The tourists go there, they think it's pretty. There's a lot of protection in the Florida Keys, so there's tons of fish. So the tourists go there and they see the reef, they see all these colorful fish, and they're like, oh, it's beautiful, it's beautiful. They don't realize that the foundation of that ecosystem is gone, it's dead. And as the years go on and bioerosion and this slowly flattens into sand, all those fish will leave and all the invertebrates that have lived there for generations. So right after undergrad, in grad school, I bought my own sailboat and I moved aboard. I lived aboard Blue Heeler. That's my little Australian cattle dog, hence the name. I lived aboard Blue Heeler for 14 years. I did grad school in Miami. I lived in a harbor that was so close to our molecular pick up Wi-Fi from the cockpit. Did a 3-year postdoc at UNAM to Florida, and I started a C school called Science Under Sail. We would take 10 to 12 undergrads and grad students over catamaran. We would do 2 to 3 week livable expeditions. We would sail all up and down the Exuma Archipelago. We would survey a bunch of the Bahamas National Trust Park and isotopes, all kinds of stuff. So I combined my 2 passions. I did this for 6 years. We would teach them all navigation. Each student would have a role for the day, and those roles would change. You were captain one day, you were navigator the next day, you were dive ops, research lead. And about day 10, I would be hands off and the students would take care of every aspect of the expedition. It was pretty cool. We graduated 54 students from this program, and all of them said that the experience was transformational to them. And several of them went on to pursue careers in marine science. So when River got old enough, he would come with me. We'd sail Blue Heeler over from Florida with all the gear. And then we transferred to the catamaran. And then afterwards, we would spend, I don't know, 3 weeks to sometimes several months just sailing around the Bahamas. So he had a childhood very similar to mine, developed the same love for the ocean that I have. So back to this colony. This colony is a massive star coral in Elizabeth Harbor, and that's in Georgetown, Exuma. It's one of the most popular destinations for cruising sailors that overwinter there. So they come to Georgetown for the winter, and then they typically sail south in the summer. I am very familiar with this coral. I probably visited it hundreds of times over a decade. I took genetic samples from it, did photophysiology on it, did 3D photogrammetry on it, and we took a 36-inch skeletal core and did stable isotopes for nitrogen in the core to show the impact of the cruising sailors in the harbor. There was this big controversy every year when the cruisers would go there, and the locals are like, "You're polluting a harbor, you're killing our reefs, and pumping out your sewage." And the sailors are like, "No, no, no, your wastewater treatment plant in Georgetown's defunct. It doesn't properly process the water." So the reefs just outside of the harbor are in much better shape than the ones in the harbor. So with that coral core, in a coral skeleton, like a tree, there's rings. And you can differentiate the summer from the winter in the skeletal bands. So we pulled nitrogen out of both, delta NT, and we looked at, okay, what's nitrogen over time in the last 30 years, and what's the difference between the summer and the winter? And that blip we saw every winter, that was the component of the cruising sailors. And it wasn't really that much, but it did promote, it prompted the park to implement a pump out service for the sailors. So this colony, I mean, it was literally a rock star. And unfortunately, in the 2023 bleaching event, it went the same way as Carey's Fort Reef. So my colleague told me it was pretty much dead, except for a few square centimeters of tissue. Very sad. This is a similar colony that I monitored during undergrad. It's about the same. I guesstimate it's probably 500-600 years old. This is Orbicella fabulata. This is 1996. In the 97-98 El Nino bleaching, it completely bleached. 2000, there was some live tissue left. And then by 2005, it bleached again. And then it was pretty much completely dead after that. So these massive corals that have been around for generations, they're gone in a relatively short period of time. I'm sure some of you have experienced this, but it's sad for me, not just as a scientist. I mean, it's sad because, these were my childhood playgrounds, and now I have a son that feels the same way. And we're watching these ecosystems collapse in a relatively short period of time. This has been highlighted in numerous publications. Half the Great Barrier Reef. The Great Barrier Reef is the largest ecosystem on the planet. It's the most productive ecosystem on the planet. In a relatively short period of time, we've lost half, and depending on who you ask, how much we've lost of that. So pretty disconcerting. So now in Hawaii, we are geographically located pretty good. We get a lot of upwelling. We are a high latitude reef system. So we've only experienced 2 mass bleaching events. One in 2015, and then one again in 2019. And we have very good diagnostic data. My boss, the director of the Global Center, Global Discovery and Conservation Science, he does all the airborne observatory. So he does spectroscopy from the airplane. He's mapped a lot of the reefs in the Caribbean, those in the Virgin Islands, and he's got about 20 years of data on the reef systems in Hawaii. So we have really good diagnostics. This is not Hawaii. This is American Samoa, but this is the same bleaching event. This is the Catlin Seaview Survey. So this is 2014, before the same bleaching event that hit us in Hawaii. And then 2015, I mean, everything's stark white. And we saw the same thing in Hawaii. In 2015, we lost about 30% coral cover in West Hawaii along the Big Island. And then 2019, it wasn't as bad. And so based on our data, our last round of surveys, some of the reefs that were bleached and experienced mortality in 2014 have actually increased coral cover. There's a small net increase. So what that tells us is if we give these systems at least 4 years, they can recover. But as bleaching events increase in frequency and severity, those windows, that frequency gets shorter and shorter and shorter, and they can't bounce back. So that's why restoration comes in. Whoops. I think this was 2 years ago, and I just really like the fact that Time Magazine had the gall to call this the most important. They didn't say one of the most important places on Earth. They said the most important place on Earth, and I don't know. I think that's pretty cool because to me it is. So, with respect to the restoration, sexual production of corals engineered for restoration at consequential scales. Again, what I'm going to talk about is in the title. But what do those two things mean? So engineered for restoration, how do we engineer corals and what do we want to engineer them for? This is a IPCC global temperature change scenario. I think it's from the AP6. And as we all know, temperatures are increasing. And even the best case scenario is still really bad for corals. If we go to the middle, the yellow and the red, it's pretty much game over for corals. So how can we incorporate something in our restoration strategies that helps prepare for what seems inevitable? And then secondly, at consequential scales. So this is and when I say Hawaii, I'm referring to the Big Island. Hawaii it's confusing. People refer to Hawaii and they refer to the archipelago, but we're just focused on the Big Island, which is Hawaii. So this is the West Hawaii coastline. It's 193 kilometers. Those are all of our survey sites, and our restoration is focused on that entire reef system. So it is the largest reef system in the archipelago. That length is larger than the circumference of Maui, so it's a huge area. So restoration has been done successfully on very small scales. But how do we get it up to something that's consequential, like the entire coastline of West Hawaii? Now I'm going to go way back and just talk about corals a little bit. You guys all know what corals are? Are they an animal, are they a plant, or are they a mineral? Well, they're kind of all three, right? Because the animal, when it bleaches, can't live for very long without the plant, and the skeleton is very important. So, but yes, you're right. They are an animal. These are some close-up. These polyps are about 2 weeks old. We settled on some substrates, then we inoculate them with symbionts. So the brown coloration you see there and there are alga within the family Symbiodiniaceae, and there's literally thousands of different species, all with different physiologies. So interestingly, I did a lot of work in the Caribbean as well, and a lot of restoration. The corals in the Caribbean are a lot different than the ones in Hawaii. All of the corals that we're working with in Hawaii, they transfer the symbiont to the egg. I don't know that there's any corals, not brooders, I don't know if there's any broadcast spawners in the Caribbean that are vertical transmitters. Are there? I don't think so. I don't think so. All the ones that I've worked with, so when the gametes come out and fertilize, they don't have symbionts, so they're required to take those up from the environment. But those symbionts, that gives the fuel, that's the food source for the animal. So when a coral bleaches, it loses a large proportion of that symbiont, and the symbiont can provide up to 100% of the nutrition for the coral. So when the coral loses that food source, essentially they starve to death. So when a coral is bleached and when it's pure white, it's not dead. The animal's still alive. So this coral this is a variation in bleaching. This part of the coral is severely bleached. The green you see, those are the fluorescent pigments. They serve as kind of like a sunscreen. And then the brown you see, that's a high concentration of endosymbionts, anywhere from 2 to 5 million symbiont cells per centimeter squared. So there's a lot of algae in there that are photosynthesizing and translocating all that sugar to the host to feed it. Again, time, nature's most beautiful death. A lot of times when corals bleach and the brown algae are gone, you see all the fluorescent pigments. So it's really beautiful, but these corals are on their way to dying. It's really pretty, but it's not a good thing. Okay, so how do we engineer corals for restoration at consequential scales? This is our approach in Hawaii. We are performing in vitro production of coral larvae on a massive scale. So this is how we plan on overcoming the challenge of scale. A coral, this is a Porites, this is a Passalopera. None of the Caribbean corals are the same as the Pacific corals. But a coral this size, even in the Caribbean, it can produce over 1 million gametes when it spawns. So that's like 1 million chances to produce a new coral. So our raceways are packed with these. This is, the Porites are hermaphroditic, so the same colony will release eggs and sperm. And then the Porites are gonochoric. You have separate sexes, male or female. These are embryos undergoing the first rounds of cell division. So once we fertilize, we watch and we look for what's called cleavage. So we'll see one cell dividing into two, two into four, four into eight, and then we can estimate how successful our fertilization was. In 14 hours, we have swimmers. So unlike the Caribbean, it takes 2 to 3 days, and you have to be very careful and rear the embryos and the larvae. In Hawaii, it's pretty simple because after you fertilize, you come in the next morning, you have swimmers, and they're what we call competent. That means they're ready to settle. So they're swimming around looking for some substrate to go down, metamorphosize, and begin the first polyp that ultimately can grow into that massive colony in 500 years. So overcoming the challenges of scale, I told you that a volleyball-sized coral can produce over 1 million gametes. We can fit 20 to 30 of those corals in our raceway. We have 72 raceways. So 20 times 72 times 1 million, I don't know, the numbers get really, really big. So that's how we are going to overcome the challenges of scale. We're not focused so heavily on optimizing fertilization. I know that there's ways that you can get, 80% instead of 60%, or even optimizing settlement. It's just we want to go with big numbers and just accept the losses and the non-optimized conditions of the process. So once we have swimmers, what do we do with them? If you dump them out in the ocean, just like natural spawning, most of them are going to either get swept away by the currents or settle on unsuitable substrate. So one of the techniques that's been done in the Philippines by a researcher, Peter Harrison, who I worked with at the University of Virgin Islands a couple times, he's doing what's called targeted larval settlement. Put these mesh enclosures down in the reef, and then they put like 400,000 larvae in this 24 square meter enclosure. That's, you can see a concentrated bag of larvae. And they force them to settle. The mesh constrains them, so they're settling in massive numbers. Because no matter what, corals are going to experience major mortality over the first several months. So by putting them in at such high concentrations, at the end of 9 months, I think they got around 250 settlers inside one of these enclosures. And then at 9 months, they had about 90 survivors. And after 9 months, no more mortality. So in one enclosure, 24 square meters, to get 90 corals that could ultimately survive to adulthood, that's pretty good. The problem that I see with this is it's also hard to scale. It takes a lot of divers, a lot of manpower to go and put these things down on the reef. So when I got to so I worked with, these are very respected members of the community, and some of them are traditional Hawaiian fishermen. So I was originally thinking, okay, why can't we use like a cast net and where the weights contour the bottom, and we just put the larval payload in like a dissolvable bag. So this was the first prototype we tested, and it got named Pololia. There's several Hawaiian elders watching, and When it went down the first time, it didn't float properly, so I zip tied a pink pool noodle to it, and they were all laughing that it looked like a jellyfish. So polio is the Hawaiian word for jellyfish. So this was the first attempt. We tested these in the pool, and it didn't really span out enough. So Uncle Paul, who's that big guy that threw the net, he's an Opelu fisherman, and they use a technique. I think they use this net system that has a rigid, they used to be made out of bamboo, now they use fiberglass rebar from Lowe's. But they make this big ring with a net and a weight on the bottom, and they send it down, and then they chum for the Opelu, and these huge schools come in, and they pull the net up. After seeing the problems we were having with the first prototype, Uncle Paul's like, well, I think an Opelu net would work just upside down. And it actually did. The second prototype we built, we spent all weekend, we hand stitched about 400 weights to the perimeter of this net. And then the pink is the fiberglass rod that actually gives it its structure. And there's some space in between the net and the rod, because when the rigid ring settles, the net can still fall and contour the bottom. And we went out and tested this, and it worked quite well. This is a long video. I'm just gonna. So it falls weird, but all you have to do is kind of like pull the top of it and settles on the reef. And then I dove down and looked at the perimeter, and there's very little space. It contoured, even those big rocks, it contoured really well. So this year we have 40 of these net systems. We still don't have an adequate payload technique. I tested 2 different polyvinyl alcohol bags. The first one, the dissolve time was perfect. It dissolved in about an hour, which is what we want, because we'll package these 200 to 400,000 larvae in these bags and then send them to the field. The problem with the polyvinyl alcohol is the first bag, when it dissolved, it got sticky like glue. So the larvae would stick in it. The second one didn't do that, but it dissolved in like 5 minutes, which doesn't give us enough working time. So we're working with some material scientists. We're looking at maybe using cellulose. This year, we're just going to have divers go down and put the payload in like Peter Harrison does. But this is scalable. We can deploy a lot of these. Paul said that a good Hawaiian fisherman should be able to deploy about 20 of these in an hour. So if we get our payload bags correct, we can give them 2 or 3 hours to go deploy the nets, attach the bag underneath. They have floats on them. We leave them in place for about 4 days. Everything should have settled by then. That's going to settle. And then we go out and retrieve the nets and make any repairs that they need. Oops. So the Kupuna were asking about recognition. They wanted to patent this and all that. I'm like, oh, that's not really how science works. I said, wouldn't you be happy if this does work, if other geographies use this technology based on traditional Hawaiian fishing practices? And they're like, yeah, but we want some recognition. So I came up with a logo. This is their village, and we put stickers on every net. And that was enough for them. They were very proud of that. So engineering corals. So how do we get over that temperature problem? So if you take 2 corals that are thermally sensitive, you make larvae, you settle them, we get the next thermal event, and the adults become stressed. But selective breeding has been used for 10,000s of years in agriculture. And it's pretty straightforward. You take, just with corn, if you have a corn population over here that's drought resistant, and you have one over here that's pest tolerant, and you reproduce, you breed the 2, more than likely you're going to get drought resistant, pest tolerant offspring. So instead of just mass producing larvae, we are testing colonies for thermal resilience. And we will take those thermally resilient colonies and we'll do selective crosses. So we'll take gametes and we'll cross thermally tolerant with thermally tolerant. And then we also do some weird ones, because there's been some research that shows it's not only the two thermally resilient ones. Sometimes if you take two thermally sensitive colonies, you get a really thermally resilient colony, but most of the time it's what you would expect. So we are implementing selective breeding into our reproduction protocols. And this is just a graphic. You take the two tall trees, pretty soon you're going to start getting populations with all tall trees. So we're going to put that in. We're going to take two thermally tolerant colonies, reproduce them, settle them. Hopefully they grow into adults and we have a thermal event and they are thermally tolerant. We're doing targeted larval enhancement. We have such good diagnostics and data on the reef systems. We know currents, we know nutrients, we know sedimentation. We have all of these data, so we can really target where we want to put these populations so that when they naturally reproduce, we can predict where their larvae are going to go and try and settle to kickstart the natural process of recovery for these reefs. So my vision is scaling this up, mass producing tons of larvae that are thermally tolerant. We're also doing assisted gene flow. We're taking colonies from the north and from the south, species that could reproduce, but geographically they would never come in contact, but we can bring them into the lab, we can cross them, and then distribute them at different sites. So there's been some population genetic studies showing that there is very infrequent connectivity between Hawaii and Maui. So if there's genetic connectivity that far away, then certainly it could happen along the coast. So that's been one of the hurdles trying to overcome. People don't, they think genetic engineering, genetic modification, but I'm trying to get away from, what am I trying to call it? I like the word genetic. So assisted larval flow, we're just taking babies one place and putting them in another. So those are the two strategies we're incorporating to increase genetic diversity, increase thermal resilience, and do it at a scale that's consequential. So my vision is traditional Hawaiian fishermen lining up in outrigger canoes. We give them their payload bags and nets, and they just go out to the reef and go crazy. Now, mahalo. Thank you.
Thank you, Dr. Robin. If anyone in the audience has any questions, feel free to raise your hand and ask away. And people in the audience online, if you have any questions for Dr. Smith, you can write them down in the chat. Anyone in the room have any questions for Dr. Smith?
Are you just working with the two species right now?
We're working with 4 species. Yep, and they're the foundational species. They're the framework builders of most of the reefs on West Hawaii.
And how many different genotypes do you have?
We don't know. We're not doing molecular genetics on them, but they're coming from far enough. That's 193 kilometers. So we're taking colonies from the north, conspecifics from the south, and we can pretty much ensure that they're not clones. Yep.
You're working with 4 species from Hawaii. This question to both of you. What are our framework species here?
We're working with 9 different species that we're monitoring for coral spawning. So that's our number. 9. Yeah. And we have validated for the captain.
I would say Orbicella is your framework species here.
I'm sorry.
I would say Orbicella is your framework species, one of the main ones. Anyone else? Yes.
How do you determine the tolerance, the thermal tolerance?
Good question. So we do 2-week temperature ramps. So we put the adults in temperature systems and we kind of mimic a natural bleaching event. So if our mean is 26 degrees, over 2 weeks, we'll ramp up to like 28 and then hold. And then we look at photophysiology. So we look at the health of the symbiont, and then we also look at the health of the animal. We can do non-destructive bleaching measurements through reflectance. So basically, we are looking at how reflective the skeleton is, because the less algal symbionts you have, the more reflectivity you have. So we can do that without harming the animal. And then we also will back test the offspring. So the larvae, we'll take a subset of larvae from our selected crosses, we'll settle them, and then we'll also submit them to temperature experiments to see if we got what we expected we would get. And then we keep refining that process over and over again. Anyone else? Awesome. All right. Thank you, guys.
Thank you so much. Great, thank you so much for that. And I am going to check, or Gail, can you check if there's any questions in the chat, please? Okay, thank you very much. All right, so now we are going to move forward. Give me one. All right. Sharing house. 1 minute. All right, perfect. So now I have the pleasure of introducing a key element in everything coral spawning that we do here at the Marine Park. I would love to introduce Carolina Rojas. She is our Reef Restoration Coordinator, and today she will be talking a little bit about our organization, our work, the program, these activities specifically, and then we'll be presenting some updates. So welcome, Carolina.
Thank you. Good afternoon, everyone. So as Andrea mentioned, my name is Carolina Rojas. I'm the Reef Restoration Coordinator, and I'm also in charge of the assisted sexual reproduction of corals. And today we'll be talking about Spawn Watch season 2025. Oh. Okay, so the presentation outline, we'll be talking a bit about Rata Marine Park, who we are, a bit of what we do, our programs, and the Spawn Watch season, which we'll mention monitoring dates, our support, and our achievements as well. So Rata Marine Park is a nonprofit organization who was built in 2005. So we have been here for a couple of years, as you can see. We're in charge of creating an environment, teaching others how to take care of the reef, how to make sustainable choices. We're here to make and empower communities as well. That's our main purpose here on the island. And how do we complete that here on the island? Well, we have some programs. We have 4 pillar programs. As you can see, we have the patrols, marine infrastructure, community development, and research and conservation. Today, we'll be talking about a bit more about research and conservation, but I'm gonna make a little small introduction into what patrols are. As you can see, some of you might have spotted our boats outside in the ocean. Our rangers are the ones who prevent any illegal fishing. They help out with any rescue situation they need, or if some boats are having trouble, they also assist them to bring them back to shore, or to help them out in any situation as well. Marine infrastructure. We're in charge of 95% of the marine infrastructure here on the island. As you can see, I tell people, like in a funny way, that we make the roads of the ocean. Just as we place out channel markers, we also place buoys, and we also place swimming zones. So with the buoys, we prevent boats from anchoring onto our reef. Channel markers is like the roads, telling boats where to go, preventing them from any collisions into our reef as well. And swimming zones is for people also safety needs to be far from the boat traffic. Community development. So in this program, we empower community. We teach them how they can use their natural resources in a sustainable way as well. And we teach them how to work out with other communities and bring out the best out of them. And for research, we have the components of asexual reproduction and sexual reproduction. We'll be talking about it. Okay, much recap. And with that, I would like to show all of you our Funwatch season for 2025. So this year we'll be monitoring for 5 consecutive months. It's gonna be a lot of fun in the diving night dives as well. So we're gonna start from May all the way to September. We're going to focus on roof break corals, pillared corals, alcorn, mountainous, and lobestar corals. As well as you can see on the side, we have mentioned which months we'll be doing night dives and what dive shops will be helping us out. As well this year, well, last year I got the support of four dive shops. This year we got 5. We got West Bay Divers, Reef Gliders, Sun Divers, Rattan Divers, and Sandy Bay Divers, who will be helping us out throughout the entire Funwatch season. And you might see that in June 19 to 22, Groove Brain Coral will be having 2 dive shops, West Bay Divers and Sun Divers, since we'll be monitoring 2 dive sites at the same time. So we're gonna be able to collect a lot of baby corals out in the reef. So this is the support of SACA, as I mentioned, of a community. We all know what's happening out there. We all come together when it's taking care of a reef. So working with these dive shops and being able to monitor 4 coral species, it's actually amazing that we can be able to bring this into the island and be able to focus and also do fertilization efforts. So now I leave you back with Andrea. She's the one who is going to be doing a bit of the introduction of the larval propagation for 2025 and a bit of a surprise for you. Okay. You're welcome.
Thank you so much. So definitely a big part of the season is the monitoring activities, everything that happens in the water. And we would like to formally invite every single one of you here. We know that you're all divers. If you're here, you're likely very passionate about the ocean and the reef. So we would like to have you all join us in the water for this magnificent event. And additionally, I would like to comment on our specific project that we're going to be doing with what we collect from these efforts. I would also like to take this moment to briefly introduce key people in this project. I would like to have Dr. Elora Lopez-Nandam stand up, Lisa Larkin, Dr. Rebecca Albright, and also Jennifer Keck, all part of the CORDAP project. Please stand up and let's give them a round of applause. Trying to go a bit incognito, but they just got here. We're very happy to have you here. Welcome back. So we're going to be talking about what the project that we are working together through CORDAP is really all about. And basically what we're going to do is conducting assisted fertilization efforts for these species that Carolina mentioned. And we're going to try to induce all of them and have them undergo 4 different experiments with the objective of enhancing survival in these bottlenecks that larvae experience when we do fertilization. So here is a brief description of the 4 interventions that we're going to be subjecting our larvae to. And all of this is going to take place in a recent facility, something that we're going to share with you now. And everything that we have on this slide will make a little bit more sense when you see what we're going to present now. All right, I think there's no audio, so I'm going to be walking you through this. So you're going to be seeing the start of this huge project. This is back in 2022. There wasn't anything built in our backyard. And as you can see, we're starting to build something that unknowingly has housed many creative minds, many creative thinkers, critical thinkers, the youth, people that care about the ocean, people that love the reef, people from all over the world that have visited this facility. However, back in 2024, it now has started to shape into what it was always meant to be, which is the Coral Restoration Center of Roatan. As you can see here, there's some footage of everything that is now part of our facility. This is not yet completed, but you'll see a lot of the progress. Those are some pumps that we're very happy about and that are going to be pumping in seawater into our facility. We were able to acquire all of this equipment that you will see there, and we will show you a little glimpse of the inside of the facility. However, it's not yet completed. Very, very soon, we will be hosting an inauguration event for this specifically, but so far, a lot of progress has been made. This is what now that palapa that I showed you earlier on looks like. And all of this has been made possible thanks to Robin and Sanka. As you saw there, they have been working extremely hard these past few days to bring this to reality. So a round of applause for them because they're really grateful. So we're extremely happy to present this. This is not only something that is ours as Roatan Marine Park, it's everyone's here. As you know, the reef is all of ours, and we hope that through this facility, we're going to be able to increase the likelihood of survival of our larvae. And now I would like to introduce, well, you already have been seeing us, but these are going to be some of the faces that you're going to be dealing with in the water. Myself, I will try my best to be in the water as much as I can. Carolina, she is pretty much a little polyp in the water. She's also going to be in the field, whether it's on our south sides or on our north sides. And then recently we are welcoming Velvet Teo. She's a very experienced instructor, has been working on the island for many years, and we are certain that she's going to enhance our project. Now I know that I introduced them briefly, but this is the CORDAP team. This is the co-PI team. As I introduced them, this is Dr. Rebecca Albright. She is the lead investigator in this. She also founded the Coral Regeneration Lab, works at the California Academy of Sciences, along with Dr. Elora Lopez Nandam. She is leading the thermal stress experiments as part of our CORDAP project, also key. We have Jennifer Keck, Education and Research Coordinator here at Roatan Marine Park, has been key in achieving all of the objectives that we have proposed and ensuring that we have adequate communication with everyone involved. And then I'm also part of the team. Now, that is what we have for you all. We are happy to be able to present these snippets. We would like to invite all of you to join us on the big events that we're going to be having in June. We're going to be formally inaugurating the facility. We will have a lot of special guests and visitors. And soon what you saw here will materialize into our CRCR facility. Does anyone have any questions? Yeah.
I have more of a comment. You very humbly said that you're also emergency, but Andrea has been leading all of the work from here in terms of liaison and contractors and all of the different pieces, and none of this could have happened without her. So.
Thank you, thank you. It's been a huge effort. All right, so anyone else have any questions? Is there questions in the Zoom? If not, I think Carolina, oh, yes, Ms. Here.
Just quick, if you did want to volunteer to go on one of these spawning dives, how do you do so?
Yes, Carolina has two lists that I'm sure she mentioned when you walked in. There's an attendance list and then there's a registration list. If you give us your contact information, we'll be reaching out to you to share dates and the logistics, where we're gonna go, when we're gonna go out, times, all of these different things. So if you have not yet signed up for that, please do so. And we also encourage you to please follow us on social media. That's where we share a lot of our updates and these types of opportunities. I don't see any questions in the chat. Okay. Okay, can you read them here? Or I can't see them. Oh, never mind, yes, I can. All right, so Javier Masso has a question for Dr. Smith. Do you also implement directed evolution of endosymbionts?
No. Because all of our,
Sorry.
all of the species we're working with in Hawaii are vertical transmitters. So the eggs come out with symbionts, and it would be almost impossible to try and expel them and put in new ones. I did a lot of that in the Caribbean. We would inoculate aposymbiotic settlers with different species, Durstinium trenchi, which is thermally tolerant, and other species to see what the outcomes were. But we're not doing that in Hawaii because of the way that they vertically transmit into the eggs.
Thank you. And then there's another question. How do you choose the depth where to deploy the nets?
Well, we have a very good diagnostics of where the degraded reefs are. So the depth really doesn't matter. We target our sites based on currents and based on kind of like need. We want to restore the natural breeding populations in certain places, so that's where we target the level enhancement.
Thank you. All right, I do not see any more questions. We'll give a few minutes if anyone else wants to say anything or in the chat. But if not, I would like to thank all of you for being here. Thank you for making this event happen. And now for all of you, as a thank you for joining us, we have some refreshments and some snacks at the back. So please feel free to help yourselves, and anyone in a blue shirt can give you more information about the organization, about our project, or answer any questions that you might have. Now, a big round of applause for all of you, and thank you very much for being here. All righty.